Okay, so some topics for review. So going way back to like the first chapter that we talked about homeostasis. And I know that's a word that you've all heard in your other science classes, but particularly we talked about two kinds of feedback mechanisms, a negative feedback mechanism and a positive feedback mechanism. In a negative feedback mechanism or loop, some change is corrected or put back the way it was. So, for example, if the body gets too hot, that triggers a response that cools the body back down. So we call it a negative feedback because it negates or reverses whatever the stimuli is. So if it's too hot, the body cools down back to zero or back to neutral. If the body is too cold, it heats up. So it's opposite. The direction of change is opposite the initial change or the inciting change. Positive feedback mechanisms are a little bit weirder because a small change is amplified. So we call it positive because it's adding to the change that already took place. So in blood clotting, for example, once blood starts to clot, it clots even more and even more and even more. So it's like an amplification. So those are those two primary feedback mechanisms. The example we gave for the negative feedback loop was the air conditioner. You know, when the room gets too warm, the air conditioner turns on, cools the room back down, and then the air conditioner turns off. Most of the feedback mechanisms in the body are the negative version, because the body has a particular way it wants things to be. So anything that departs from that, and the body's going to do something to correct it, to bring it back into that normal range. All right. And then we talked about anatomical position and directions. So anatomical position is standing palms forward. You know, it's not exactly the normal way we stand. You know, usually we stand with our palms turned in. But in anatomical position, it's upright and palms forward. And we talked about the directional terms. You're going to use these not only in this course, but in the rest of your medical career too, to describe things. So just as a review, we have anterior and posterior. Anterior is towards the front, posterior is towards the back. Now, because the human being stands upright instead of walking on all fours like a lot of animals do, anterior and posterior have other words too. So anterior is also ventral, or towards the belly, and posterior is also dorsal, or towards the back. So if you remember that a shark's fin, the jaws fin, that's the dorsal fin of the fish, and that will help you to remember that dorsal is in the back, on the back of the animal. All right. Lateral and medial. Lateral is away from the midline of the body. Medial is towards the midline. And since the human being is symmetric across the midline, we have a left half and a right half that are pretty much the same, at least on the outside. We use these medial and lateral terms a lot to describe, going further from the midline or going towards the middle. Proximal and distal we use when we're talking about the limbs in particular. So proximal is towards the origin of the limb. Distal is toward the end. We also use these words when we talk about vessels and nerves. So arteries and veins, we'll talk about, things, a vessel is distal to another vessel or proximal to another vessel. So proximal and distal. And then the set that is used least commonly is that cranial caudal one. So cranial is towards the head, caudal is towards the tail or towards the end of the spine. Now in other animals, we use cranial and caudal a lot more because they are on all fours, so we can describe it easier that way. But for the human, only rarely, and usually we talk about cranial and caudal when we're talking about the central nervous system. Because the brain is at the top and the end of the spinal cord is at the bottom, so these words make sense in that context. All right. And then the planes, the body's a three-dimensional thing, and sometimes the best way to think about whatever anatomy you're looking at is to think of it in one of these three planes. So the transverse plane is the one that you actually see in clinical medicine the most often, and this is cutting a person in top and bottom, so their top half and bottom half. And the reason that we see these most commonly is the computed tomography, the CT scan, and the MRI. Because of how they work, the pictures that they generate are usually in the transverse plane. So anybody who's going into hospital-based medicine or ICU, nursing or OT, transverse plane is something you'll get familiar with, but not in this class. So that's top and bottom. If we cut the person in left and right, that's a sagittal plane or a sagittal section. The down the middle into left and right, so we're going to divide the person in half. We call that the mid-sagittal plane because it's the middle sagittal plane. So we get pictures that look like this. And then if we cut the person into front and back, then that's the frontal plane. Now, most of the pictures in anatomy books are usually rendered in the frontal plane. You're usually looking at the person head on. You just have to remember that the anatomy is more complicated than that. We're not South Park characters with no third dimension. We do have a depth, and organs are not, evenly distributed from front to back. So the three planes, sagittal, frontal, and, all right, let's see, why is my mouse acting up? There we go. Okay, so the planes. All right, so that was chapter 1, at least in quick review. Also in chapter 1, we talked about words and word roots, how anatomy words are like Legos. You take 3 or 4 different words and you can put them all together to make a compound word. Hopefully you're starting to see that. And you'll see it more as we get into the organ systems and we start talking about things. So like the epidermis, we've talked about that. Epi is above, dermis is skin, so this is the top part of the skin. So just an example of a Lego word. All right. Then we talked about chemicals. We did a little chemistry review with a particular focus on how chemistry matters to the human being. We talked about water. Water has a number of important properties that made it the medium of life, so to speak, or all living things that we've ever discovered have water involved in their physiology. Some of the reasons for that: water is known as the universal solvent. Now, it can't actually dissolve everything, but it can dissolve lots of things. So because water can dissolve so many different things, it allows complex solutions or mixtures of substances dissolved in water. So it has a high solvent potential. Water has a high heat capacity. In other words, it takes a lot of heat to increase the temperature of water. And that matters because by being water-based, it means that our temperature has a tendency to stay put. If we were, mostly alcohol, for example, alcohol's heat capacity is much lower. So when it got cold outside, we'd get colder faster and we would get hotter faster. So high heat capacity. And then water is polar. Now this is a way to describe a chemical, but it means that it has a positive end and a negative end. And the reason that matters is polar substances can dissolve in other polar substances. So because water is polar, it means that it can hold lots of different ions and substances that have one end or another. The other version is nonpolar, and in the biologic molecules, it's only the lipids that are nonpolar. All the others are polar. All right, so water. We talked about carbohydrates. Carbohydrates, their primary role in the body is as nutrients. So the most important carbohydrate to know is glucose. Glucose is a monosaccharide. It is a simple 6-carbon ring. It's the simplest sugar, and it is one of the primary fuel sources for the body. The mitochondria, they love ATP, or they love glucose, and they make lots of ATP from it. So glucose is a primary energy molecule. If we string a bunch of glucose molecules together in kind of a branching pattern, we create a starch called glycogen. So a starch is a long chain of simple sugars, okay? And our starch, the starch that the body makes, is called glycogen. We make it out of glucose, and it's a big branching chain. All right, so carbohydrates, fuel molecules. Lipids, three primary functions in terms of lipids. One, phospholipids create cell membranes. And we learned that there are multiple organelles in the cell that are effectively made of membranes. So these phospholipids are critical to life. And you'll remember that the cell membrane is called a lipid bilayer, because what we get is we get the phospholipid heads all line up on the outside, and the tails all point towards the center. And we make a three-dimensional structure out of this, a sphere, and that's the cell membrane. So each one of these, so one of these guys, is what we call a phospholipid. So it has a polar head, and it has a non-polar tail. And because of that, it orients like this almost by itself. So it creates the cell membrane. All right. So that's one important lipid. Another is triglycerides. So by mass or by weight, most of the lipid in the body is in the form of triglycerides. Triglycerides are the primary component in adipose tissue, so in fat tissue. And what a triglyceride is it's, so this is a carbon backbone, so each corner is a carbon molecule. And a triglyceride is this 4-carbon chain called glycerol, and then attached to it are these long chains of carbon and hydrogen that we call fatty acids. So a triglyceride is made up of 3 fatty acids and a glycerol backbone. And it's triglycerides that our body makes for energy storage. You know, when you take in more calories than you spend, the extra gets saved up as triglyceride. And then also, because triglyceride is a good insulator, it also helps to keep us warm, and it helps to cushion and protect things. All right, so phospholipids, triglyceride. And then the last one is cholesterol and the steroids, which are chemically related. Cholesterol, and I'm not going to draw it accurately because I can never remember how, but it looks something like this. So cholesterol is a joined ring. So it's multiple rings of carbons that are all sort of joined together. Cholesterol's primary role in the body is in the cell membrane. It lives in the nonpolar region of the cell membrane, so in between the heads where the tails are pointing together, cholesterol lives in there. And it helps to keep the cell membrane very flexible. And then cholesterol is made into certain kinds of hormones called steroid hormones, which directly affect the nucleus. So all that to say, cholesterol and its sister, the steroid hormones, are the third important lipid that we see in the body. All right, so phospholipids, triglycerides, cholesterol. Then we talked about proteins. Proteins are long chains of amino acids. And an amino acid is just a kind of molecule that has a nitrogen on one side and a carbon on the other. But what we end up with is an unbranched long chain of amino acids. So this is amino acid 1, this is amino acid 2, amino acid 3, and so on. All right. The bond between 2 amino acids, we call this a peptide bond. And it's called that because it's a nitrogen bond. Peptide is always associated with nitrogen. So because these amino acids have a nitrogen that connects them, they get this special name called a peptide bond. So this long chain of amino acids is not going to do anything in this form. You know, a long chain isn't very interesting. You know, it's not going to do anything exciting. So proteins have a shape that they develop as the long chain kind of twists and bends and loops around itself to make a kind of blob. You know, if you imagine a long string that you take and you kind of ball up, that's sort of what a protein looks like. You know, the long string gets turned into a shape, and then that shape has effects. So we talked about 3 or 4 levels of structure for the protein. So its primary structure is the order of amino acids. And that order of amino acids is what is stored in DNA. You know, DNA is a recipe for making a protein, for putting a string of amino acids together in just the right order. So that's the primary structure that comes from the DNA through the RNA. The secondary structure is if we take this long string and we do something simple to it. You know, let's say we twist it into a helix, or we line it up in kind of a flat sheet, you know, like you would do for weaving. That's a secondary structure. And then the tertiary structure is a single protein's final shape. Okay, so we go from the order to a simple pattern, and then we take that simple pattern and we make it complicated. And that's the tertiary structure. Without the tertiary structure, the protein doesn't work. It doesn't do what it's supposed to do. And then the last one we talked about was quaternary structure. There are a lot of proteins that are made up of more than one part. So like hemoglobin, for example, has 4 separate protein parts that are joined together. So the quaternary structure is multiple proteins that are joined together into one functional unit. And there are a lot of important proteins that are like that. All right. So a little review of proteins. Ions are atoms or molecules that carry a charge. And they're very common in the body, and they're very important in how the body works. So a cation is a positive ion, and an anion is negative. So cation, and the common mnemonic there is the cation has a positive in the middle, right? There's a T in the middle. And then anion, a negative ion is an anion, just little mnemonics. And there are examples of both. And we talked a little bit about the common ones found in the body. So sodium, potassium, chloride, bicarbonate, we talked about all 4 of those. Calcium is a major player in terms of ions. But for now, just know what an ion is. It's a molecule with a charge. All right. pH is our measure of acidity, and what pH actually measures is hydrogen ion concentration. So, you know, the simplest atom in all the periodic table is the hydrogen ion, the H+, because quite literally it's just a proton. It's a single subatomic particle without an electron, without a neutron. It's simple and boring. But because it's simple like that, it's reactive. So the more hydrogen ions you have in a solution, the more reactive that solution is going to be. That's acidity. You know, if you take something very acidic and you put like metal in it, the metal will bubble and eventually dissolve because those hydrogen ions are literally taking the metal apart. So the pH is a measure of that hydrogen ion concentration. Now, pH matters a lot in the body. Because of that, the body tries to keep it in a very narrow range. The reason for that is proteins change their shape as the pH changes. And if the protein changes its shape, the function of that protein changes. So the body defends a pH of 7.4. So 7.4 is normal body pH, and it departs from that very little. And we briefly talked about buffers. Buffers are substances that, when present, reduce the change in pH. So if you have a beaker with water in it that has a buffer in it, and you add acid, the pH doesn't change until you add enough acid to use up all the buffer. And we're going to talk more about this next semester, but just know that pH, hydrogen ion concentration, and that buffers help to keep the pH from changing. Okay. We talked, and then our last sort of biological chemical we talked about was DNA and protein synthesis, which I know that you've heard in other classes in your past. So, it shouldn't be a total review, or a total new thing, rather. But DNA stores the recipe for making proteins, but proteins are made out in the cytoplasm. DNA is in the nucleus, right? So we have a location problem. The recipe is in one place, but the machine is in another. The kitchen is somewhere else. So that's where RNA comes in. RNA copies the recipe, takes it out into the cytosol, and then attaches to a ribosome, and then that ribosome creates a protein matching that RNA recipe, which matches that DNA recipe. So a couple of important words that sound similar but are different. Transcription is the creation of RNA from a DNA template. Okay? We call it transcription because we're transcribing RNA language to DNA language, which are not very different. Translation is turning that RNA template into an actual protein. So translation is another name for protein synthesis, and transcription is another name for RNA synthesis. And you'll hear those words come up. So transcription is DNA to RNA, translation is RNA to protein. All right. And then we started talking about the cell. Cells don't exist in the body all by themselves. They're always connected and associated with other cells. And the connections between those cells can be critical to how a cell works. So 3 I want to remind you about. The gap junction is a connection between cells that allows stuff to pass through. All right, so we have cell. So here's one end of cell 1, here's another of cell 2. So a gap junction is like a little straw between the two. So there's a hole here. So it creates a channel or a path so that molecules that are small enough can actually go from one cell through this tube to the other cell without having to leave and come back inside. So we see gap junctions in tissues where the cells have to work together, or they have to do something all at the same time. Probably the most famous example is cardiac muscle. Cardiac muscle has these gap junctions so that the whole heart will contract at once, so that the signal from this cell can pass directly to the next cell without having to go out and come back in. We also see gap junctions in smooth muscle, like we see in the intestine or the uterus or the bladder. Same reason. All those smooth muscle cells have to do the same thing at the same time, or the whole system doesn't work. All right. So that's a gap junction, like a straw between the two. A desmosome is like a rivet or a nut and bolt. So on one side, we have a kind of protein plate. And then on the other, we have the same thing. So just underneath the membrane, there's like a disc of protein on each side. And then those discs are connected together by a very strong protein. So desmosomes keep cells from being pulled apart. You know, if you grab this cell and you try to pull them apart, those two plates are going to resist that, you know, with the binding in between. So desmosomes give cells strength against shearing or being torn apart. So like our skin cells that we talked about, they have lots of these, you know, because the skin cells have to hold on to each other. Otherwise, if you rubbed your skin, well, the cells would just all rub off. So that's a desmosome. And then the last one that I wanted to talk about is called an occluding junction. Okay. So again, we have cell 1 and cell 2. An occluding junction is a little bit like a stitch in clothing. It sort of weaves in and out like this. And what it does is, because these cells are sort of stitched together, stuff cannot get between the cells and into the body or into the tissue. So we call it an occluding junction because it blocks the path between the cells. And an occluding junction looks a little bit like a stitching pattern. All right. So those are three I wanted to remind you about. Okay. Organelles. So we talked about, it's just like the body, the cells have structures that have particular function. The nucleus runs things, and it does so by controlling what genes get activated, or what genes. A gene is an area of DNA that is the recipe for a protein. And because the nucleus has control of all the recipes, it has control over what gets made in the cell. So the reason we think of the nucleus as being in charge is it determines what the cell does by turning genes on and off, essentially. So creating a protein or not creating a protein is going to alter what that cell does. All right. The endoplasmic reticulum is a site of manufacturing. So the rough ER is where proteins are made. The smooth ER is where everything else is made. So things like phospholipids and amino acids, things like that, are made in the smooth ER. So endoplasmic reticulum manufacturing. Mitochondria ATP production. Specifically, mitochondria. So if this is a mitochondria, they look a little bit like that. If we put small carbon chains in, so we put small carbon chains in and we put oxygen in, and we get ATP out, we get CO2 out, and we get water out. So essentially, the mitochondria is a factory that burns carbon chains into CO2 using oxygen and then forms ATP and water in that process. So the reason that we breathe is because of the mitochondria and what they do. We have to bring in oxygen and we have to get rid of the CO2 that has been formed by burning the food that we eat, or the nutrients that we eat. So mitochondria are responsible for that. And then ribosomes are protein factories. So they use a messenger RNA template to create a chain of amino acids, which then goes on to become the functional protein after it reaches its tertiary or quaternary structure. All right. And then we finally got started talking about the human being. So up until that point, we had been talking about, general chemistry, general biology, and then we started talking about tissue types. So remember, all of the tissues in the body are one of those four types. So it's either an epithelium, connective tissue, muscle, or nervous tissue. There's only four types. Now, inside each of those groups, there are different tissue types inside there, but those are the four big categories. We talked a lot about epithelium. Epithelium, one of the things to remember is it's considered polar. In other words, an epithelium always has a top and a bottom. The top we call apical, and that's going to point either towards the lumen. Okay. And then the bottom we call basal or basilar. So top and bottom. Epithelium always sits on a membrane, so the basilar side sits on what's called the basement membrane that sort of gives the whole epithelium a structure. Epithelium is either lines something, in other words, covers the surface of a thing, or we find epithelium that form glands that produce a substance, like sweat, for example. Another place we see epithelium is in senses. So like, we have a sense of touch thanks to what are called neuroepithelium, or epithelial cells that respond to stimuli somehow. So most of our senses actually arise from epithelial cells that are specialized for that. So like your eyes, your ears, your sense of taste, your sense of smell, your sense of touch, all of those are because of epithelium. Okay, so we talked a lot about epithelium. Connective tissue, the big characteristics there are there's a lot more stuff than there are cells, and that stuff we call the matrix, which is made up of protein fibers and then ground substance, which is sort of the fluid part of connective tissue. And we describe connective tissue as loose or dense, and then regular or irregular. And you should have looked at some of these in lab, too, the different tissue types. Now, there won't be any questions on our lecture test about identifying specific tissues. That's for lab. But in terms of characteristics, like which kind of tissue has a top and a bottom or an apical and basilar side, well, that's epithelial tissue. None of those other tissues have that. So do know the characteristics, but you won't have to identify a tissue from a description, for example. All right. So tissue types. And then we actually got started talking about an organ system, particularly the skin. The skin has two primary layers, right? There's the epidermis on the top, and then underneath the epidermis is the dermis. And we divide the dermis into two layers. The papillary layer sits just underneath the epidermis. And this is where the blood vessels that supply the cells of the epidermis run. The papillary layer gets its name. Papilla means hill. And the papillary layer has all these hills. And the epidermis has a matching hills, so to speak. And that helps to keep the epidermis and dermis attached to each other. Because there again, if this were flat and smooth, when you pushed on the skin, it would have a tendency to separate from the underlying dermis. So instead, we have this kind of egg crate pattern that holds those epidermal and dermal layers very tightly together. So papillary layer. And then the larger layer underneath of the dermis is the reticular layer. And this is where you're going to find the fibers that make the dermis strong and elastic. So we have collagen fibers that give the dermis its strength. And then we have elastic fibers that give it its stretchiness, that the skin will stretch and then snap back, like when you smile and then frown. The skin can do that because of the elasticity of the dermis. So papillary layer and reticular layer. Underneath the dermis is the hypodermal space, which is not technically part of the skin. It's under the skin. But this is areolar tissue, like we've all seen in lab. And it allows the skin to move separately from what's underneath it. Okay, so layers of the skin. And then if we zoom in to just the epidermis, so we're going to look at just the top part, we will see, we'll see the layers of the skin, or of the epidermis rather. There are two kinds of skin. There's thin skin and thick skin. Most of the body is covered with thin skin. The only places that are covered with thick skin are the walking surfaces. So the palms of the hand and the soles of the feet are the only place that we have thick skin. And we have, in thick skin, we have 5 layers. 1, 2, 3, 4, 5. In thin skin, we don't have this one. So we only have 4 layers. And just to review, the stratum, let's use my pointer here instead. All right. The Stratum Assaleh, which is right here. This is the stem cell layer. So this is where the mother cells are. These mother cells divide, and one cell becomes a daughter, and the other stays a mother. So it can continue to replace the cells above it. So that's the Stratum Assaleh. The Stratum Spinosum is the largest layer of living cells. So we have kind of layer after layer of keratinocytes. Remember, these are keratinocytes. That's what we find in the skin. So that's the Stratum Spinosum. The Stratum Granulosum is a thinner layer that lies here at the top, and you can see it in this picture. It's kind of blue. And we call it granulosum because it has granules in it. So when you zoom in under the microscope, what gives this area a darker color is the cells have a bunch of granules or little grains in them. And those grains contain keratin, which is then going to create this top layer that we'll see in a minute. In thick skin, we have this layer called the stratum lucidum. Lucid is clear, so this is the clear layer. And you can sort of see it here, how there's a clearing. Not so much here, but you can see it here, and here, and here. So that's the one that's different. So stratum lucidum, only found in thick skin. Okay? And then above that, we have this the stratum corneum. And as you've seen under the microscope in lab, this layer can vary in size a lot. You know, this is thick skin, obviously because it has a lucidum. So the palms and soles have just layer after layer after layer of these dead keratinized keratinocytes. So just huge numbers of layers. If you look at the scalp or, you know, the skin from the hand, you'll see many, many fewer layers of stratum corneum. So like it might even only be like as thick as this, you know, right here, this little part. So huge variability. The stratum corneum is made up of dead keratinocytes and the keratin that they ended up making. So all the while these cells are moving up, because they start down here and they end up here, as they're moving up, they're creating more and more keratin, and eventually they die in the granulosum layer, and then what remains is the keratin that forms a kind of flexible, waterproof shell for the human being.
Now, do those dead layers that are just keratin, those still have melanin, or is the melanin underneath?
No, they still have melanin.
Okay.
Yeah, they have the dead ones have melanin too. Yeah, and these guys are sloughing off all the time. So like up here, you know, we all know we lose skin cells. We don't lose live skin cells. We lose dead skin cells from the stratum corneum all the time because new ones are being made. You know, the skin's always replacing itself. All right. Some accessory structures of the skin. We talked about hair a little. We talked about nails a little. But there are 3 glands that are important to be able to discriminate in the skin. So we have 2 kinds of sweat glands. One is very common. The other is found only in specific areas of the body. So when you think sweat gland, what you're usually thinking of are the miracrine sweat glands. These are the sweat glands that produce a very watery solution that has a little sodium chloride in it, a little salt in it. And their purpose is to cool the body down. By putting a little water on the skin, and that water evaporates, it takes heat away with it. So it's a kind of evaporative cooling mechanism. The miracrine sweat glands also are a little bit antibacterial. So it makes our skin a little less of a nice place to live for potential bacteria. All right. The other kind of sweat gland is found in only certain areas of the body, so axilla, groin, and nipples. And these produce a very viscous secretion, so not watery at all. It's thick, it's kind of slimy, it's very oily, and it has a lot of complex chemicals in it. Now the thinking is that these apocrine sweat glands are probably pheromone glands, so olfactory communication, sending signals from one organism to the next. Because they're very influenced by hormones, they don't develop until puberty, so there's some thought that these are pheromone glands for the human being. These are the ones that are responsible for body odor. Miracrine sweat glands don't produce anything that bacteria can use for nutrients. The apocrine sweat glands do. So the bacteria take the secretion, and in their metabolism, they end up releasing gases that have a smell. So body odor is associated with the apocrine sweat glands, not the miracrine sweat glands. All right. And then the last gland is the sebaceous glands. Sebaceous glands are oil glands that basically help to keep the skin nice and supple and nice and soft. You always find them associated with hair, so every hair has a sebaceous gland related to it, and that's because without that oil, hair gets very dry and brittle, so it breaks. But the sebaceous gland helps to keep that hair flexible and soft so that it can do its job of protection. But we also have sebaceous glands scattered around our skin as well, so they're not all associated with hair, but all hair cells have an associated sebaceous gland.
Are we going to need to know the difference between a gland and a follicle?
No.
Okay.
No. A follicle is just a place where a hair is.
Okay.
Okay, I think that's, let's see. In terms of osseous tissue, the stuff we just covered, there's compact bone and there's spongy bone. Compact bone is arranged in osteons. Osteons being sort of concentric cylinders. Compact bone is very strong and hard and heavy. And then spongy bone is arranged in trabeculi or sort of a complex strut network. It's less strong, but it's better at transmitting forces in multiple directions. The main cell types, osteoblasts make bone. Eventually, the osteoblast surrounds itself with bone and becomes an osteocyte that lives in a lacunae, or a little gap in the bone for the cell to live. Osteoclasts dissolve bone, and then I guess that's the three. Yeah. Osteoclasts dissolve bone. Bone is always remodeling, so it's always being dissolved and remade to keep bones as heavy and strong as we need, but not heavier or stronger than we need. And I think that's kind of all I have on there because we just talked about it. So those things are less important.
Are we going to need to know the flat bones?
Yes, the different kinds of bones. Yep. The classification system. All right. Okay, so I spent most of our time here, but let's just do a couple of questions here. All right. So get your soccer thing out, and I have everybody's name. There we go. Okay. So what is a tissue? No, it's not reliable enough. That's why I have a sheet up here that I do it on. Our old clicker system, you could do attendance like that, but no, it's new ones. All right. All right, I think that's everybody. Okay, good. A tissue is a group of cells with similar structure and function. Important to note that an organ is always made up of more than one tissue. Okay, so all four tissue types, epithelial, connective, muscle, and nerve, are found in all organs. So don't confuse organ with tissue. Tissue is part of an organ, but it's a set of cells that are all doing the same thing. All right, what's an organelle? Ooh, I'm glad I asked this question. We got a diverse set of answers. All right, jump in there, last couple of people. All right, there's my 24. All right, so an organelle is a cell organ. So it's a, let's see. It's I can see why there was a spread. A small structure within a cell is not a very specific definition, but you can say that's an organelle. Because a structure composed of several tissue types, that's going to be an organ. Basic structural unit of all living organisms, that's a cell, right? And group of organs with a common set of functions, that's an organ system. And we already talked about E as a tissue. So kind of a weak definition for organelle, but once you've eliminated the others, it would have to be A. All right, not the greatest. We're going to skip that one. Okay, this is a good one. Which of the following is most similar to a negative feedback mechanism? This one you have to think a little about, which is part of why it's in here. All right, 5 seconds. Jump in there. Got to commit to an answer. Good. Unlike classes in the past, most of you got this one. This is C would be the best answer here. All right, so why? A negative feedback mechanism works to put a thing back the way it was. You know, so before you flush the toilet, it was full. After you flush it, the tank empties. So a negative feedback is, well, we want to put it back the way it was. We want to fill that tank back up. A car runs out of gas and stops. There is no feedback there. You know, that's just running out of a thing. So now if you stop to get gas because the car was out of gas, you have participated in a negative feedback loop because you have put the car back the way you want it with a full tank, right? The same is kind of true with B and E. There isn't a feedback there. B would actually be a neutral feedback mechanism because all the teacher is doing is identifying the situation. It's not responding at all. D would be an example of a positive feedback mechanism where the response to a change is to make something happen, different happen. You know, normally the door is closed. When you approach it, the door opens, so that's a positive response. You've made something happen, not put something back. All right, we'll do, actually, let's call it right there, because we only got a minute left anyway. So I'll post the review questions, obviously, that we didn't get to online. I'll open up all the clicker questions that we've had so far. Homework you'll be able to review after midnight when it's due. And then I'll see you all on Friday for our first test together. Do study, but don't panic.
And you said that you were going to change that homework?
Yeah, I'm going to look at that.
So it'll be out of 9 instead of out of 10?
Yes. Yeah. Which screws up the percentages, but it doesn't really matter.